Having anything personalized to your best interest is always optimal. Vital health care is no different. That is why the idea of not being treated for a disease, but by your disease, is a concept that is increasingly popular with both the researchers and cancer patients. In order to push this personalized care, there must be a more in-depth understanding of both normal and cancer cell biology. To speak on this subject is veterinarian turned cancer development biologist, Dr. Aimee Edinger.
So in here we have leukemia cells growing, prostate cancer cells, colon cancer cells, breast cancer cells, and this allows us to study the different cancer types outside of the body, outside of a mouse or a human.
And you said you use these because the cancer cells grow more rapidly than regular cells.
Right. So in order to do the research, we need to make lots of cells to be able to analyze them. So cancer cells are great for that. If you took a normal cell and put it into one of these dishes, it would divide a few times and then stop. It would do something called going through senescence. And senescence is like an aging process, but it can happen on a cellular level. So the cells just kind of flatten out and stop growing if they're normal. But the cancer cells continue growing forever. They're what we call immortal.
Which is part of the problem when you actually have cancer. So what is it about the action of cancer cells that makes them grow more rapidly than other cells?
So just the cells have lots of checks and balances inside of them to make sure that they don't grow when they shouldn't, and when they're damaged, they die for the good of the organism. And cancer cells have disruptions in all of these checks and balances. So rather than, when a normal cell is damaged, for example, by UV radiation or a chemical carcinogen, then the cell can sense that there's something wrong with its DNA. And the normal response is for the cell to go through this process called apoptosis, or programmed cell death. And that's from the Greek terms that mean leaves falling off trees. So it's a cellular process where the cell kills itself for the good of the organism. And that's what a normal cell should do when it's damaged. But the cancer cells, they circumvent this pathway. They find ways to evade death, and they stay on in the body even though their DNA is damaged. And those mutations that let them survive death can also cause them to proliferate out of control. You might be surprised to learn that every single cell in your body has 6 billion base pairs of DNA. That's an incredible amount of DNA. And every time it divides, all those bases have to be copied exactly. Right? And so there's a DNA polymerase, an enzyme that makes new DNA from the template, the old DNA. And even if that makes a mistake 1 in a million times, if you're copying 6 billion base pairs of DNA, every time a cell divides, there's going to be a few mutations that don't get fixed during that DNA copying process.
So DNA damage happens, whether it's genetic or some sort of external environmental force. And then now these DNA damage starts turning into a cancer cell.
Right.
Like you said, these cancer cells like to grow more rapidly. At what point do they start turning into a tumor or start actually growing?
That's a really good question. So in the lab, we can make a cancer cell by mutating or damaging 4 different genes. But in the real world, usually there's maybe 20 to 30 mutations that happen before a cell progresses all the way to cancer. And you know that there's early stage cancers, right? And then there's late stage metastatic disease, and all the time the cancers are accumulating more and more mutations.
Do all cancers metastasize or grow at the same rate, or are they all different?
No, certainly. There are some kinds of tumors that are less likely to metastasize and some that are more likely to metastasize. And a really important area of research is finding biomarkers, finding tags on those cancer cells that tell us, is this going to be a metastatic cancer cell, or is this going to be one that stays in the primary tumor? Because most of the time you can treat the primary tumor. It's the metastatic lesions that are hard to deal with. Probably are familiar with the term personalized medicine.
Right.
We can sequence genomes so easily. You can sequence the genome of the cancer cells as well and find out what changes have happened in the cancer cell that aren't in that person. You can follow the genetic changes over time in the DNA. And we're hoping that as sequencing the genome becomes cheaper and cheaper, we can devise therapies that target the exact mutations that you have in your tumor.